to thank Emmett and the organizers for having us here today. We are a publicly traded company, so I draw your attention to our forward-looking statements. We're listed on NASDAQ. So at Stealth, we're focused on mitochondrial dysfunction. We have a number of programs, late-stage rare genetic mitochondrial diseases. We have a program in phase 2B for dry age-related macular degeneration, and we have a robust pipeline with the first of our pipeline assets moving into the clinic later this year. So again, from a pipeline perspective, later stage in rare genetic mitochondrial diseases, I'm going to focus you today on our dry age-related macular degeneration program, where we're enrolling our phase 2B clinical trial. So Elamipretide is our lead compound. It binds preferentially to cardiolipin, which is an important structural component of the inner mitochondrial membrane. Why do we care about that? Mitochondria produce about 90% of the energy in our body, and when they become dysfunctional, whether due to inherited genetic mutations or the aging process, and you can think about risk factors for disease like AMD, such as cardiovascular health or obesity or smoking, which are highly oxidative stress related, mitochondrial dysfunction can ensue, driving the production of more and more reactive oxygen species in the body. The inner membrane of the mitochondria is curved. Those curves are called cristae. They look more like tubules than the prior illustration showed you. And that curved architecture of the inner mitochondrial membrane is mediated by cardiolipin, which, with mitochondrial dysfunction and increased oxidative stress across different diseases of aging, can become quickly peroxidized, which leads to a breakdown of the inner membrane, a drifting apart of our electron transport chain complexes, less ATP production, and more oxidative stress. Across multiple preclinical models, you're looking at the retina from a diabetic mouse. In the middle, you can see that cardiolipin breakdown, which resulted in a loss of inner mitochondrial membrane in the mitochondria. On the right, with a 6-week course of treatment, Elamipretide normalizes mitochondrial morphology, and really that cristae is interconnected typically across networks, shown in yellow. In disease, it becomes disconnected and dysfunctional, restored again with Elamipretide. AMD is characterized by mitochondrial dysfunction. You can see very characteristic lack of cristae architecture, lack of inner mitochondrial membrane in the disease, so a good functional target for a drug that targets mitochondrial function. Based on strong preclinical data showing potential improvement in vision in animals, as well as dry AMD donor eyes, we did a phase 1 study at Duke, enrolling 40 patients with intermediate dry age-related macular degeneration. Elamipretide is dosed systemically once daily in the abdomen. It's a multi-use cartridge and pen similar to an insulin pen. These patients were screened for a 5-letter low light deficit. Half the patients had drusen, the other half had non-central geographic atrophy. We hope to see an improvement in visual function in these patients over the 6-month course of treatment versus what would expected to be an up to 5-letter decline in that period. So looking at low light vision, and on top is going to be the drusen cohort, and on the bottom will be the geographic atrophy cohort. We saw about a 5.5-letter gain in vision in both cohorts. For patients, the 14 patients who had already progressed to wet AMD in their non-study eye, they also experienced a 6-letter gain in low light visual acuity on top of standard of care. Regular light vision was not that impaired in these patients, so the 3.5 and 4.5-letter gains we saw brought them back to about 20/20 and 20/25 vision, respectively. 5.5-letter gain in the non-study wet eyes. We asked patients to read the smallest font size possible in 30 seconds through a dark filter and saw significant improvements in low light reading acuity. We saw improvements across multiple parameters of visual function, including particularly peripheral and color vision, which, being rod and cone mediated, suggests that the drug may be improving photoreceptor function. In terms of geographic atrophy growth, this is compared to the natural history over a 6-month period. It does appear that Elamipretide is reducing the rate of geographic atrophy below what would be expected, measured both by FAF and OCT. These reads were done by Duke and Burke. So our phase 2b clinical trial, 180 patients focusing on geographic atrophy, dosing for 1 year to hopefully amplify the separation we saw on geographic atrophy. Primary endpoint of low light visual acuity, which has been approved by FDA. We've discussed this data with them. It's 2-to-1 randomization, currently enrolling at 40 sites in the US. We're targeting full enrollment early next year, which will get us data early 2021. So pleased to move this program, obviously, from early animal models, translational benefit we believe we saw in our phase 1 trial, where we saw an improvement in multiple parameters of visual function, and now enrolling the phase 2b with data expected early 2021. Thank you.